In the age of wellness, it's tempting to believe that there's a perfect diet, a universal supplement, or a golden ratio of macronutrients that will lead us all to the promised land of longevity. But the truth is messier and far more interesting. The future of health isn't about rigid food pyramids or one-size-fits-all advice. It's about personalized nutrition, a model that accounts for the wild complexity of our genes, gut microbiomes, and medical histories. And few nutrients better illustrate this complexity than niacin. Niacin, vitamin B3, essential for energy metabolism and cellular repair, plays a starring role in the synthesis of NAD, a molecule involved in everything from mitochondrial function to circadian rhythm and DNA maintenance. It's also a therapeutic agent used at high doses to manage cholesterol and triglycerides. But here's where things start to diverge. Not everyone responds to niacin the same way, and for some, what's healing in one context may be harmful in another. Let's begin with genetics. Certain people carry variants in genes related to NAD, metabolism, lipid transport, or detoxification, meaning their ability to convert niacin into usable NAD or process niacin's metabolic byproducts can vary dramatically. For instance, polymorphisms in the NQO1 gene, which encodes a key NAD, H dehydrogenase, can affect how efficiently cells recycle NAD. Likewise, MTHFR mutations, often discussed in the context of folate metabolism, can interact with niacin metabolism through shared methylation pathways. Individuals with these genetic quirks may require more or less of certain B vitamins, or benefit from different forms, like nicotinamide riboside, NR, or nicotinamide mononucleotide, NMN, which bypass specific metabolic bottlenecks. Now layer in the gut microbiome. It turns out your resident microbes can synthesize niacin themselves from tryptophan or directly from dietary precursors. But the quantity and quality of this production depend on which microbial species are present and how well they're nourished. A fiber-rich, plant-diverse diet might support niacin-producing microbes, while a high-fat, low-fiber, or ultra-processed diet might starve them out. Dysbiosis, a microbial imbalance common in inflammatory diseases and metabolic syndrome, can also impair niacin synthesis or lead to accumulation of toxic niacin metabolites like 2-PY and 4-PY, which are associated with kidney stress and vascular inflammation. In other words, your niacin needs may rise or fall depending on the microscopic organisms you're hosting. One person might need extra niacin because their gut isn't producing enough. Another might need less because their microbiota is pumping it out like a factory. And in some, high-dose niacin could trigger a cascade of pro-inflammatory metabolites if the microbial machinery isn't tuned correctly. Then there's the wild card: personal disease history. Have you ever had fatty liver, diabetes, kidney dysfunction, or cardiovascular disease? These conditions can fundamentally change how nutrients are processed. People with insulin resistance, for example, may respond to high-dose niacin with worsening blood sugar control, a phenomenon well documented in large trials where niacin increased the incidence of new-onset diabetes. Similarly, individuals with renal insufficiency may be unable to clear niacin's byproducts effectively, increasing their risk of toxicity even at moderate doses. And in people with low-grade systemic inflammation, niacin might not have its intended anti-inflammatory effects at all because chronic inflammation can suppress NAD synthesis pathways or upregulate competing metabolic routes. What all of this points to is a seismic shift in how we should think about nutrition, not as a set of blanket rules, but as a dynamic, personalized system where nutrients interact with your unique biology. It's no longer enough to say niacin is good or too much is bad. Instead, we need to ask for whom, under what conditions, in what form, and when. This is where nutrigenomics, microbiome sequencing, and metabolomic profiling come into play. These tools are rapidly evolving, allowing clinicians and researchers to map how individual genes and microbial species influence nutrient metabolism. Imagine a future where your niacin needs aren't guessed, but calculated based on a blood test, a stool sample, and your personal health history. Your optimal diet might include moderate niacin intake with evening meals, prebiotic fibers to support niacin-producing bacteria, and perhaps a niacinamide supplement post-workout to support mitochondrial recovery, while someone else might thrive on a completely different protocol. Until then, personalized nutrition begins with self-awareness. Track how you feel with different foods and supplements. Get your labs checked, especially if you're taking high doses of B vitamins. Talk to a provider who understands that normal ranges may not mean optimal for you. And remember, longevity is a long game. What works for your neighbor might backfire for you, not because the nutrient is good or bad, but because your biology is uniquely yours. In the end, the path to health isn't paved with generalized advice, but with precision. The better we understand the intricate interplay of our genetics, microbes, and history, the closer we get to a future where nutrition isn't just prescriptive, it's personalized, preventative, and profoundly powerful.